In Module A for Objective 12, we're going to look at differences between the skeletal muscle that we've been looking at and other muscle types, specifically cardiac here. Cardiac muscle, if you remember from lab, is striated, and it's only found in the heart. Now, this striation should bring something up that we've just covered. What caused the striations in skeletal muscle? You should be thinking about the organization of the sarcomeres and those thin and thick filaments. So we can anticipate, since cardiac tissue is also striated, that we could expect to see some similar type of organization within the cells here. In looking at cardiocytes, we can differentiate them from skeletal muscle in a number of ways. Cardiocytes are relatively small. If you remember, the muscle fibers are these multinucleated giants, some of them reaching up to 12 inches in length. Cardiac muscle cells are not. They often, in fact, only have a single nucleus, though on rare occasions you might find a cardiocyte with more than one nuclei, but nowhere near the numbers we find in skeletal muscle. The T-tubes, the T-tubules of the cardiac muscle cells are relatively short. They're wide, and we do not find the formation of triads. In fact, the sarcoplasmic reticulum, though present, does not form those large chambers, those terminal cisternae. The cardiac muscle cells are highly aerobic, and because of that, we find a specialized protein located in cardiocytes called myoglobin. And this is very closely related to hemoglobin. And of course, most of us know the function of hemoglobin. It's a protein that binds and carries oxygen. Myoglobin has a very similar function. So this is a way to make sure the heart has the oxygen necessary for aerobic respiration. You can imagine the importance of keeping the heart fed. Obviously, we cannot allow the heart to go without its necessities due to the importance of this organ. And we also have a tremendous amount of mitochondria. Again, the heart needs, it beats throughout your life, and it needs a supply of energy. Another characteristics found in cardiac muscle cells, you may or may not have seen these in lab, depending on the quality of the slide, are the intercalated discs. And we'll talk more about the function of these structures later. In fact, right now. Intercalated discs are these specialized connections between cardiocytes. They actually join the cell membrane of one cardiocyte with the cell membrane of the adjacent cardiocyte. And within the intercalated discs, we're going to find structures such as the desmosomes and the gap junctions. So this is a way of connecting these cells together. And they function to maintain structure. They enhance and conduct action potentials. And this is probably largely the more important characteristic of the intercalated disc. When you have cardiocytes contracting, they all have to contract at the same time. When a signal is sent, all cardiocytes must contract so that the blood can be then pumped or ejected out of the heart. So these intercalated discs play a role in allowing that electrical signal to be transmitted from one cardiocyte to the next. And it happens very quickly throughout all cardiocytes, which allows all those cells to contract at roughly the same time. This is a diagram, and we'll blow up some of these pictures. Up at the top, we have a histological prep of cardiac tissue. Directly underneath that, we focused in and have a drawing of what a cardiocyte would look like. Here again, we have the single nucleus. We have a cell that is branching, and we also see the repeating striated patterns, which again should let you believe that the actin and myosin are arranged in a very ordered fashion, as we saw in the skeletal muscle. And this bottom photo is basically looking at how the sarcoplasmic reticulum and the T-tubules are organized. This kind of increases the size of those two figures. Cardiac tissue, it's striated and it's branched. Here we actually have the intercalated discs. Very nice capture of those intercalated discs. And as we look at that third figure, again we have a sarcoplasmic reticulum. That's this blue network, and it's wrapped all around the protein, the actin and the myosin. And we have these very short, thick, or wide T-tubules that run from the sarcolemma as it wraps around the myofibrils. What's missing here? Well, we obviously don't have the terminal cisterna, and because of that, we also do not have the triads. So, intercalated disc and the coordination of cardiocytes. Because of the intercalated discs, the heart itself acts as a single, and the textbook will call it fused mass of cells. It acts as a single unit. And again, it's very important that all those cardiocytes contract at roughly the same time. So this is achieved by these intercalated discs. We have four functions that can be found or can be associated with cardiac tissue. Automaticity is one. This is contraction without a neural stimulation. The actual electrical activity of the heart is controlled by the pacemaker, and this is a group of neuron-like cells that maintain the electrical activity within the heart, the pace. And this can be demonstrated. We've all probably seen this in movies or something where the heart is ripped out of the chest and it continues to beat even without a signal from the central nervous system. And that is because the heart itself has its own signal. Cardiac tissue has variable contraction tension, and this is controlled by the central nervous system. Along with that, we have extended contraction times. Contraction time in a cardiocyte is 10 times as long as that of the skeletal muscle. Cardiac tissue also has a long refractory period, and this is due highly to prevention of wave summation. In Objective 13, we're going to look at the differences between smooth muscle and skeletal muscle. Smooth muscle is found in areas like the blood vessels, the lining or the walls of the blood vessels, reproductive and glandular systems and organs. They're found in the digestive system as food is pushed through the digestive tube or the digestive system. It's largely accomplished by smooth muscle. Found in the urinary systems as well as the integumentary system. And let me just remind you of the erector pili muscles that ultimately give rise to goose flesh or goosebumps. Skeletal muscle is non-striated. It has actin and myosin, but it's organized very differently from what we have seen thus far. And that's going to give rise to different functional characteristics as well. This is a histological prep of smooth muscle. we have here both longitudinal and transverse sections. If a skeletal muscle cell, smooth muscle cell is often described as being spindle or cigar shaped. And if you were to slice that in the longitudinal plane, it would look something like that. And if we were to look at these cells, we could almost pick one out using our imagination. But if we were to cut this transversely, what you would end up with is something that may or may not look like that. And again, that's what the type of cut that we have over here, where we have a transverse cell, a transversely cut cell showing only the end point. So those are the differences, and it often confuses students because students, especially in histology, they don't look or think in 3 dimensions. But you really need to consider how that cell is cut. The actinomycin are, as I said, they're organized very differently. We do not have the striated patterns that we find in skeletal and cardiac muscle tissue. The actinomycin are present, but they're just organized differently. And they tend to be bound by the structure known as a dense body. And we'll talk more about dense bodies a little bit later. This picture here looks at the relaxed form. You can see the cell has its normal spindle shape, and when contracted, the actinomycin interact to kind of, I don't know what you would call that, shrivel that cell up. It almost looks like a crenated cell, one that has lost a lot of its water. Characteristics: spindle-shaped. They have a single central nucleus. Smooth muscle tissue lack T-tubules. They lack the myofibrils as well as the sarcomeres. They do not have tendons or aponeuroses. I think we all remember tendons. Aponeuroses are those flat, broad, sheath-like connective tissue structures that hold muscle onto bone, for instance. The myosin fibers are scattered, and the thin filaments, the actin, is found attached to those dense bodies. And it's the dense bodies that actually transmit that contraction from one cell to the next. In Table 10.4, we have a really good comparison. They're looking at properties between skeletal, cardiac, and smooth muscle. It's just a good kind of review if you wanted to go back and try to summarize all of that material.